All right, looks like it's about that time. We'll go ahead and get started now. I have 1 minute after by my clock, so we will kick it off. Hello to everybody who's on the line, and thanks for joining us today. And hello to everybody who is watching this on demand in the future. I'm so glad you're interested in this topic and willing to spend your time with us. I'm excited today to continue our new Macmillan Teaching Topics webinar series with this discussion about incorporating bioinformatics into your course. My name is Will Moore, and I work as part of the life sciences team at Macmillan Learning. We are so happy to have the chance to engage the STEM community with a series of teaching webinars, so thank you for taking the time to join us. Our speaker today is Michele Shuster. She's a professor of biology at New Mexico State University, focusing on the scholarship of teaching and learning, and she teaches introductory biology, microbiology, and cancer biology classes at the undergrad level, and also works on several K-12 science education programs. She also happens to be an author of Biology for a Changing World, which is a textbook for the non-majors biology course that Macmillan publishes. So before we get started, just a few logistical details. Because we have a large audience, voice communications are disabled, so everybody is muted. I'd encourage you to submit any questions or comments that you may have using the question function in the sidebar controls of GoToWebinar. We really want to hear from you. We want this to be as interactive as possible, and we want to be able to answer your questions. So please, I encourage you to make use of that. I'll be monitoring the questions as they come in, and we'll have time for Michele to address these toward the end of the session. So with that, I will pass it along to her to go ahead and get us started.
All right. Hi, everybody. I also want to echo Will and thank you for taking time out of your day to join us. And like Will said, he'll be monitoring the questions, and so please do chime in with questions. I'm happy to answer them. I apologize, my camera has decided not to play today, but hopefully you can, okay, so you can basically be like my students. You can listen to me just like they've been doing asynchronously all semester, but since you are synchronous, you can at least chime in with questions, and I will be happy to answer them. So as Will said, our official title is Bringing Bioinformatics into Your Classroom, but the way I've been thinking about this is DNA-based case studies. And so a couple of weeks ago, I did a presentation on using case studies to teach science. And if you weren't here, it's perfectly fine, but this is, I think of it as an extension of that, where we can start to use the tools of bioinformatics to make some DNA-based case studies. So just in case you weren't here and haven't thought a lot about case studies are basically stories. And so we are storytellers. We like to listen to stories. We like to tell stories. And so basically a case study is some kind of a story. More specifically, they are scientific stories for our purposes, for our teaching purposes. And even more specifically, as Kip Herod would say, they are stories with an educational message. So that's kind of the context around which we're going to frame our discussion today. Is we're talking about stories, but we're also talking about DNA-based case studies. And so what is a DNA-based case study? And of course, I'm sort of making this up, so this is my opinion of what a DNA-based case study is. So it's a case study in which you're going to use some open access bioinformatics tools, some DNA sequences, maybe some protein sequences. And so you're going to use these tools and these sequences to help advance your story, whatever the story of your case study is. Maybe you're going to use them to solve a mystery, or maybe you're going to use them to reinforce some of the content that you're working on with your students. The other reason why I really like thinking about DNA-based case studies is they can actually contribute to this broader societal goal. And so the National Human Genome Research Institute has this GLEE project, so Genomic Literacy, Education, and Engagement. So this GLEE initiative. And basically the GLEE initiative focuses on 3 target groups to help advance their understanding of genetics and genomics. So there's healthcare providers. So even our healthcare providers may not have had a strong genetics background, and so they can be in need of some genetics education. The general public, certainly that seems incredibly appropriate right now when we're hearing in the news about different strains of COVID and tracking where different COVID eruptions, emerged. I mean, that's definitely a genetic concept, as well as, the 2 vaccines that are being talked about right now are mRNA-based vaccines. And so that requires some understanding of the central dogma. So certainly the general public definitely would benefit from understanding a little more about genetics and genomics around the current conversations. And then the third target group are our students, the K-16 student population. And so, as I said, the K-16 students, those are our students. We're the 12 through 16, but as Will said, I also work with teachers, and so I sometimes consider the younger peeps my students too. And so we can use DNA-based case studies to really address genomic literacy. Do you understand genetics and genomics, and how can we use a case study to help reinforce your understanding of genetics and genomics? So a nice example of a DNA-based case study that really does reinforce concepts of genetics and genomics is one called Solving a Medical Mystery with Bioinformatics. And that's published in the National Center for Case Study Teaching and Science by Brackett et al. So that's a really nice example if you wanted to check out an example of a DNA-based case study that's really reinforcing sort of concepts of genetics with the students. But I would argue that we can think outside of the genetics box when we're thinking about these DNA-based case studies. Yes, we can use them to reinforce understanding of genetics and genomics and central dogma gene expression, but we can really go far beyond that. We can think of the DNA and protein sequences, we can think of those bioinformatics tools as literally just tools. So we don't have to necessarily use them to reinforce genetics, but we can use them to solve our mystery, to reinforce other aspects of science, and certainly to practice critical thinking. So we can really expand the use of these tools to address all kinds of student learning outcomes. And I don't know about you guys, but in New Mexico, we have state mandated general education essential skills, and one of those is critical thinking. And so we certainly embrace every opportunity we have to have some kind of critical thinking components in our gen ed courses. The other thing that I think is really cool about DNA-based case studies is they are literally appropriate for all levels. I mean, you might think, oh, it's DNA, it's genomics, it's tools, you know, that sounds like an undergraduate kind of activity. But we have actually used DNA-based case studies with kindergarten students. So you can go all the way from kindergarten, elementary school, middle school, high school, and all the way up to the university level. And so there are lots of opportunities. For example, if you have bored kids at home right now, you can give them a DNA-based case study to do at home as, you know, just for grins, because what could be more fun than a DNA-based case study? So one example that we've published, again, in the National Center for Case Study Teaching and Science, it's a murder mystery. It's a murder by HIV. And we have a version that we've written for basically middle school students, grades 5 through 8. But we also have a high school version of the same DNA-based case study and an undergraduate version. And so it's the same basic story, but it's scaffolded in different ways for those different groups of students. And since Will did mention, you know, working with teachers, and many of you, I'm sure, are being asked right now to help come up with ideas for teachers that they can use with online learning with their students, I just wanted to share a few that our team has developed with our NIH-funded Science Education Partnership Award. And so that SEPA grant, we worked with elementary and middle school teachers and helped them design their own DNA-based case studies to use in their classrooms. But at the same time, we've also generated, you know, a small collection of DNA-based case studies that can be used for those younger students. And so these are all on our science tools in the classroom, so stcnm.org resources. So for example, and I'm going to walk you through this one just so you get a sense of what I'm talking about once we get there. We have one DNA-based case study where students investigate what microbe killed the Aztecs during a large outbreak between 1545 and 1550. We have one that's actually based on a scientific paper where literally scientists got clumps of hair that, you know, hunters or hikers claimed came from Yeti, and then we give them DNA sequences, and the students determine the origin of those sequences and determine, you know, what in fact these Yeti sightings are likely to be. I'll give you a hint, it's not actually Yetis. We have another activity, Parmesan by any other name, where students compare the bacteria that are present in American Parmesan cheese and authentic Italian Parmigiano-Reggiano, and they can look at the sequence diversity and then make conclusions about maybe why these two cheeses might taste different. Another one called Risky Virus is about assessing the risk for the outbreak of a West Nile virus, comparing two towns. And basically what the students do is they use sequences to identify whether all of the components necessary for an outbreak are present in either of the two towns. Our federal fish one is basically, it's fishy. It's fishy business because students basically are given DNA sequences that, for example, maybe a fisherman or fisher people claim comes from this species of fish, but it comes from a different species, or lower quality fish are being passed off as higher quality fish. And so basically students determine, is that really the fish that it's purported to be? And then what are the implications? And so sometimes there's conservation implications. For example, if the fish that's being sold under a false name is actually potentially an endangered or threatened species. So again, they get into a little bit of critical thinking, and then they come up with a plan on how to prevent further violations of this sort. And we have one for younger kids where they can use DNA sequences to identify bioluminescent or glowing organisms. And then younger students might think, well, all the organisms that glow must be closely related, right? And so they can actually use an online tool to build a little phylogenetic tree to show that the glowing creatures are actually from different groups of organisms. And so they might not necessarily be all that closely related. So those are some of our examples that we've made. And if you were to go to the National Center for Case Study Teaching and Science and just use bioinformatics as a search term to search their case collection, that will call up 24 different case studies. And the vast majority of those are designed for undergraduate level classes. Okay, so I said a DNA-based case study is one that uses tools of bioinformatics. And so what kind of tools might those be? So a tool that's used in basically all of these is the BLAST tool from Ncbi. And so BLAST, for those of you who are not familiar, you can use it to compare two different nucleotide sequences, or to take an unknown nucleotide sequence and compare it to all the sequences in the database to find a match and therefore identify the source of that sequence. You can also do the same thing with protein sequences. So you can align two protein sequences and look for differences and mutations, or you can put in an unknown and compare it to the database to identify its source. So these are very common tools, and the things that certainly the teachers like and often students like are these are the same tools that real scientists use. So we're not just making up behind the scenes tools for classroom use. These are the same tools that practicing scientists use. And because it's not always intuitive how to use these tools, so for example, on our STC website, we have developed an activity that's just to learn and practice using BLAST. And so this is kind of our starter activity. So before you do anything else, go in and do the learn and practice BLAST. The one thing I will tell you is that periodically Ncbi and BLAST change their user interface. And so I do try and go in and update our learn and practice BLAST to make sure that the screenshots match the current interface. But if you were ever to hop on there and notice it looks out of date, please let me know. That is one of the downsides to using live tools, is the tools evolve or the interface may change. And then we want to do our best to keep up so that people who don't use them every day still have like a guide to follow to use these tools. So that's BLAST. Another tool we use is abbreviated MAFT, and this is a multiple sequence alignment tool. So basically you can put in a whole bunch of sequences, and then it will generate, well, it will align them behind the scenes, and then it will generate a phylogenetic tree, so a tree of relationships based on the similarities and differences of those sequences. So one of the activities that we do is trying to figure out if the panda is really a bear, because it looks kind of like a bear, but it has these not so bear-like qualities. And so we put in a whole bunch of different organisms, including sequences from the panda, and the students are able to figure out that the panda is indeed more closely related to the bears than to other kind of carnivore type organisms. So those are some of the tools. BLAST and tree building are the two most common. And so what I thought I would do, just to give you a sense of what this might look like, is to take you through the Aztec activity. So I've kind of built it just so that it stays in the slideshow in case, you know, we have internet failure or some other crisis. So it won't be completely live, but it'll give you an idea of how we put these tools together, in this case, to sort of solve a mystery and practice critical thinking. So again, this is the What Microbe Killed the Aztecs, and we developed this as part of our SEPA, NIH SEPA award. And this was actually an activity that was developed by a former grad student. So she's now Dr. Stacy Scroggs. And she designed a lot of these activities as part of being a research assistant on this project. And so basically, we're looking for what microbe killed the Aztecs. And so we give the students a little bit of background about the Aztecs. So they were the Nahuas, and they lived in what is now Mexico at the time when the Spanish were colonizing the Americas. We talk a little bit about their common language, where they lived, and just a little bit of background to who this group of people were. And then we ask the question of what happened to them. And so obviously things were changed permanently in 1519 with the bringing in of culture, language, government, religion, food, as well as disease. And so the native populations had not been exposed, and so they didn't have immunity like the incoming Europeans. And so it turns out that a lot of these disease outbreaks were incredibly serious and devastating, and killed up to 95% of the local populations. And since then, historians and scientists have tried to use descriptions of the symptoms to try and hypothesize what pathogens may have caused the outbreaks that killed so many of the Nahua people. And so again, Stacy was able to develop what I'm about to tell you based on a publication. And so we try to give sort of authentic sequences and authentic stories. And so between 1545 and 1550, there was a massive outbreak, and I'm not even going to try and pronounce it, Kokolitzli. But samples, biological samples from people who died during that outbreak were collected from cemeteries in southern Mexico. And we were able to, well, the scientists were able to analyze and get sequences for pathogenic DNA. And this was a serious outbreak. It killed 80% of the Nahuas. And there are symptoms, right, that were recorded. So fever, black tongue, yellow skin and eyes, seizures and pain. It sounds like it's sort of hard to identify what the pathogen may have been. But as the students will see, we can take these historical samples from the cemetery and compare them to DNA from pathogens that we either know have caused outbreaks in the past or we suspect have caused outbreaks, to try and figure out what is the actual pathogen that was responsible for this particular outbreak from these people from the cemetery. So the first thing that students do is they're given some reference sequences. So they're given sequences from pathogens that we know have caused outbreaks in the past. So we're giving them basically a bank of organisms that we know have caused outbreaks in the past. So we're kind of giving them a library from which the cemetery deaths, you know, might have been caused by. And so we typically use ribosomal RNA sequences or ribosomal DNA in order to do our species identification. So the first thing the students do is they blast. So they use BLAST, and they blast reference sequence A against the database, B against the database, et cetera, in order to determine what these disease-causing organisms are. And so they write down the scientific name and the common name, and they're also asked to figure out if it was a bacterium or a virus. So that's the first thing they do. And so they, hopefully, so far it works every time, they use BLAST, and so they're able to identify smallpox, which is a virus, influenza, which is a virus, measles, which is a virus, salmonella, bacterium, and then Yersinia pestis, which causes the plague, and that's a bacterium. So now they sort of have this, you know, set of organisms that we know have caused outbreaks in the past. And so then what they're going to do is be like, okay, these are the kinds of things, right, that we should be thinking about. So the next thing they do is they are given DNA samples from the cemetery samples. And so these are published in the paper. We can use their reference IDs to go in and pull out the sequence for students to blast. So they're going to blast 3 of these from 1545 to 1550 cemetery samples. So we have cemetery sample 1, 2, and 3. And when they do that blast, they find out that this is basically, it's consistent, it's all Salmonella. And in particular, it's all the Serovar paratyphi C. So it's all Salmonella bacteria, and it all appears to be the same sort of subspecies or serotype of Salmonella that caused the cemetery. And remember, Salmonella was one of the bacteria that we know have caused outbreaks in the past. So at this point, for the younger students, we might end it here and say, all right, well, what do you think caused it? Salmonella. Does that make sense? Yes, because we know Salmonella has caused outbreaks in the past. For more advanced students like high school or even undergrad students, the third part of this is we have them go in and actually generate a phylogenetic tree to compare those cemetery sequences to some modern day Salmonella sequences. And so they're going to use, again, the 3 cemetery sequences that they've already blasted. And then we give them 2 modern, like present day DNA sequences. So we call those creatively modern sequence 1 and modern sequence 2. Modern sequence 1 is the same species and subspecies as the cemetery sequence. But remember, those cemetery sequences are from organisms that were alive 500 years ago. And so this modern sequence 1 has had 500 years to continue to evolve and diverge. Okay, so even though it's the same species and subspecies, there's 500 years of genetic separation between this and the cemetery sequences. And then modern sequence 2 is the same species, it's Salmonella enterica, but it's a different subspecies. And so then we ask them before they go in and build their trees, how do they think modern sequence 2 is going to relate to the other sequences? Do you think it's going to be the closest relative to the cemetery sequences or the more distant relative? And hopefully they'll be able to figure out that modern sequence 2, which is the same species but different subspecies, is going to be more distantly related to the cemetery sequences because it's a different subspecies than modern sequence 1. So they go in and they can go to a website where they can basically input their sequences into a window, and then they hit the compute button, and then they wait, and then a page loads, and they literally click a view tree button. And then what we ask them to do is draw their tree on a piece of paper. But I mean, it shows up on their screen, but if they draw it on a piece of paper, they can kind of run around and show it to one another. And basically what they find is their tree should look like this. So you can see here are the 3 cemetery sequences, the ancient or 500-year-old sequences. And so they're clearly all clustered together and have a common ancestor here. Modern 1, remember that's the one that's the same species and subspecies as these 3, but it's had 500 more years of evolution on it. So notice it's a little more distantly related. It shares a different ancestor than these 3. And then finally, modern 2, who's the same species but a different subspecies, is the most distantly related, which is hopefully what they predicted. So this is a pretty simple tree. Even if they're not great at reading ancestors, they can kind of read clusters, which I know is not ideal how to read a tree, I understand. But for younger students, it gets them sort of started thinking about, how to read a tree and how to interpret what you're looking at. And so, for example, that Parmesan cheese activity I mentioned, it's similar to this in that they first blast a bunch of bacteria that are found in the cheese, and then they assemble a tree and establish relationships among the bacteria and compare the American and the Italian cheeses. And then some of these activities just involve blast. So they are blast-only activities, so they just identify and use that to advance the case study story. So with that, I hope that gives you an overview of sort of DNA-based case studies, and I wanted to leave a lot of time for questions or comments if people have them. So I intentionally did not want to sort of blab at you forever because I know that's bad practice, but hopefully if you have any questions or comments, I'd be happy to try and answer them.
All right, thank you so much, Michelle. We have had a few come in. Mind if I rattle them off here?
Go for it.
All right, do you use these case studies in lecture or as a lab activity?
So I personally use them in lecture, but I also know that our lab coordinator has used them in labs occasionally, or variations of them. So especially when we are in lecture talking about, central dogma and mutation and like that, she often has some kind of a DNA-based informatics type project that they will do in lab. And that's been actually really easy, thankfully, to transition to online because those tools are there. And if you have the instructions, students can generally pretty easily work through those tools in NCBI.
All right, thank you. How do you go about deploying these types of case studies in an online class?
So in an online class, I tend to scaffold them a lot more. And so I will often do like a screencast and sort of walk them through the handout in the screencast and show them the tools and the screenshots and everything. And I tend to give them a little bit more elaborate instructions. I also tend to give them a worksheet so they'll watch a screencast that has the directions and kind of works them through the worksheet. Then they do it, and the worksheet has space to kind of write notes like, what do you predict about this? What do you predict about that? And then typically what I'll do is once they've done all that work, which is I hope where they're kind of doing their critical thinking, maybe they're visiting with a classmate, maybe they're visiting in office hours to check their understanding. Then I set up, we use Canvas as our LMS, so I'll set up some sort of a quiz where they can go in and enter either open-ended text, or I might convert some questions to a multiple choice, like, who's the most distantly related to this, who's the most closely related, and then have it be a drop down or multiple choice to sort of help them make sure that they were on track.
All right. How do students react to this type of case study versus some of the other types? This is kind of getting back to the last webinar where you discussed a number of different types of case studies.
I think my experience has been students very much enjoy these. For people who were here last time, and if you weren't, don't worry too much, but I had talked about a case study where the main point was to help students understand protein localization, and they had to predict where a protein was going to go in a family with kidney disease. And the basic problem is it's a peroxisomal protein that experiences a mutation that sends it to the mitochondria. So you end up with your mitochondria clogged up with a peroxisomal protein, and that's what causes all the problems. So in that case study, there's actually a small bioinformatics part in that the students compare, I give them a partial sequence of the wild type protein and the mutant protein. They have to identify the change. Actually, I give them protein sequence. They identify the different amino acids. Then there's another bioinformatics tool that you can put in, and it basically will predict like a signal sequence or a mitochondrial targeting sequence. So from that, they can figure out if what targeting sequences are present in the protein. And then that can help them develop the hypothesis for, oh, this mutation created a novel mitochondrial targeting sequence. And then that's consistent with they observed the protein in the mitochondria on kind of micrographs. And so they really like that they could get on the computer and like do this thing and realize that just that one amino acid changes where that protein is going. And I think that can help solidify things that are otherwise a little bit more abstract. And so they seem to enjoy using the tools. When we did it with like the kindergarteners, whenever they hit blast, the teacher would have them like stand up and make like rocket ship noises, and they go, I mean, I don't even know if they really knew what they were doing, but they liked, and then they had like, oh, it's this fish or it's that fish. And then the teacher had pictures of the fish whose sequences they were blasting. So the little kids had a blast with BLAST.
Cool. How many of these case studies do you typically do in one semester?
Well, in a normal semester, like pre-COVID and pre-online, so we have like what, 14-week semesters, I think, 14, 15, whatever. I would normally have 9 of these that take up a full class meeting. And they might not all be DNA based, but I would have a case study 9 times in the semester. And it would be like a 75-minute full out in-class celebration of science. This semester, now that I'm online and doing weekly modules, there is an activity, like a case study activity, literally every single week. So I needed 12 this semester, so I had to write some extras. But then I also had to sort of re-scaffold them and make them a little more approachable, like I said, for students who are working more independently in my asynchronous environment.
Okay. Are there certain topics that seem to be bigger hits with students than others, like things about food, disease, crime?
They like crime. I have one case study, it's not a DNA-based one, but they look at both, well, they look at different things that can kill you and then figure out why, and it all has to do with cellular respiration. So they kind of liked that. I will tell you, they just finished it. Like I just finished grading it this morning. And in the normal classroom environment, I would tell them the names of the metabolic poisons. But last semester, I realized when we went to our emergency online, they were Googling and typing in all this stuff like that didn't make sense because they Googled and it didn't answer my question. So this time I anonymized the poison. So it was like poison A, poison B, and poison C. And they had to sort of work out, okay, tell me how this is going to kill you, tell me how that's going to kill you. And then today I put up on the Monday morning announcement the identity of the chemicals. And I've already had a few emails like, I had no idea cyanide did that to you. I'm like, yes, it's all about cellular respiration. So they like murder and mayhem. I have one case study that I do on doping in sports, like blood doping.
Mm-hmm.
In the classroom, that one cracks me up because we talk about both illegal and legal ways to enhance your athletic performance. And some of us are like, oh, I could do that. I'm like, I just told you that's illegal. I'm like, yeah, but I bet it works. I'm like, oh my God. So that seems to be a big one. And then of course I show them data that it actually didn't work and it killed the people. So don't go there. Yeah, I found historical stuff for my students doesn't seem to go over as well. And that may be because that's partly my bias. Like I'm more like a current events kind of gal. So I try and like rip my stuff from the headlines as it were. They like disease. They like mysteries of disease, especially like if you show them someone with, say, like a very disfiguring disease, they're like, oh my gosh, like how did that happen? What's going on there? And then they really want to know.
All right.
So that's been my experience.
Learning a lot about students and their, aspirations of being Dr. Evil, I guess.
Yes. We did have a discussion board where it's like, if you were an evil genius and wanted to cause either spastic or flaccid paralysis at the neuromuscular junction, how would you do it? Because I wanted to see if they understood like signaling. And there were some great ideas. But then I could tell them, oh, well, that's how Botox works, or that's how this anesthetic works, or that's how this thing works. And like, so they actually came up with, real things. So that was kind of cool.
Nice. All right, last question we have in here is, can you share the link for the publication of this Aztec case study?
Yes, well, here's what I can do. Let me just go back to where I had it on the bottom of some slide here. So it is on, I'm just moving this thing out of the way. So it's here, it's www.stcnm.org. And then you have to click enter, or you can just go /resources, and that's where all of these different DNA-based case studies are, these ones that I showed you. And then, like I said, the National Center for Case Study Teaching and Science has a whole bunch as well, if you just go onto their site and use bioinformatics as your search term.
Great, thank you so much for going back to them.
Sure thing. And again, let me know if one of the links is not working right, because they do update stuff and I have a hard time keeping up. So yeah, and so again, let me go, hold on. I go all the way back to the beginning. Here's my email, no C in Schuster. That's the most common reason it doesn't get to me. Drop me an email if you notice that anything needs to be corrected, and I will work on getting it fixed, or I'll just send you an updated version.
Great. All right, well, we will go ahead and wrap with that then. So thank you so much, Michele, for the presentation. I'd like to thank everybody who attended live and all those who end up viewing this in the future. And we will see you next time. Thanks a lot and have a great day, everybody.
Bye, everybody. Thank you.